So what we'll do here, and we've changed some things around, you will notice that some things have changed not so long ago. We have moved the metagenomics section right here. And what we will do today is we will just take a look at this last example called amplicon 16S 18S. So if you click on this dropdown, you will open this section right here. And this is called the data to pipeline. So you will find right here in the middle a section called data to. Now, we're not going to go into a lot of detail about this. What I want you to see today is just how to run a demo pipeline, what are the inputs and outputs, and what does it mean? Where do you go from there? So here you want to click on start. You can see what we covered today, kind of what is being sequenced. The data to pipeline is specifically designed for pair-end metagenomic sequencing data for amplicon 16S ribosomal RNA. So here, the 16S ribosomal RNA hypervariable regions are being sequenced, not the full gene. And then one of the objectives of this pipeline is to actually denoise the actual sequences. So the more precise the sequences that we have, the better we will be able to match them accurately to the reference database. And then if we have an accurate match, we can then also analyze differences in composition, or maybe how they are related to each other phylogenetically. So here, an important step is to understand the challenge, right? And one of the challenges is with this great diversity of microorganisms that we are studying, that we are sequencing, there is expected to be a lot of variation. And because of that variation, it's important for us to differentiate between technical variation, like noise in data coming as a result of inaccurate sequencing, maybe because we don't have a lot of good coverage, or maybe because there is a lot of diversity. How do we actually understand the biological diversity that we have? We have to denoise and kind of eliminate the mistakes. And to do that, we need to merge the pair-end reads and then combine them into what's called operational taxonomic units. That allows us to eliminate a lot of the variability that is inconsistent. Unfortunately, it also reduces a lot of the reads that we will have, but we are only going to be keeping reads that are accurate and could be matched to something. So once we do this, I'm going to quickly go through some of these steps. We want to assign those to the right reference in this database. And assigning them means that now I can not just speak about individual OTUs, operational taxonomic units, but I can group them by whatever level of taxonomy I was able to annotate them by. And one of the challenges is going to be the lower we want to go on the taxonomy scale, the less information we might be able to get because of these hypervariable regions and their variation, but also because of the coverage and how precise we know which one came from there. Now, from this point on, we essentially have several different tables. Those tables include a table of OTU abundance, or how much of a particular operational taxonomic unit I have proportionately in each sample, and the annotation of where does that OTU map to from the reference database. Because I have the reference database, I also know the relationship between them. How close are they to each other? Could they be grouped in some way? How far away are they evolutionary from each other? And then I can perform downstream analysis, which includes building that phylogenetic relationship with ape, and then visualizing the results with phyloseq, which is a package in R that allows us to make visualizations and all kinds of statistical analysis that then we can interpret in terms of differences between groups of samples or looking at individual samples. So once we do that and we click on end right here, we will look at some of the output files. So once you click on OK, right here you will have a folder called download pipeline output files. Okay, how many of you were able to get to this last screen that I have. Okay, perfect. Rafael, did anyone else? Okay, if you did not, again, I'll just repeat the steps really quickly. So from areas of analysis, down here, amplicon 16S, 18S, you click on this drop down, go into the demo, start, QC filtering, replication and count, pair merge, cluster consensus OTU, remove chimera, assign taxonomy, ape, and then phyloseq. So after this, you click on end, and you get the download pipeline output files. Open this up, and now you have several different things. And these are the kinds of results that you're going to be getting from a pipeline. These results will include tables, TXT files, for example, plots, like these PDF plots right here, FASTA files of actual sequences, and those are the major types of data that you will have to deal with. Okay, so as an example, right, this is going to be explained in much greater detail in the course. So when you go into the metagenomics course and you want to understand what was it that we were doing, right here you have a practical 16S metagenomic analysis lesson that you can review, understand each one of the steps, and see all of the results in one place. But before you jump in there, you want to go through all of these different sections to understand what kinds of sequencing data do we have? What are the specific ways to analyze them? And then even practice some of the basics so that you understand what is going to be in an individual sample file. And to do that, let us take a look at another example. So let's go. Now, hopefully everybody has a good sense of what we can do on the platform, just what kinds of things we're going to be doing. And here we're going to be doing something else. Here we're going to try and run a piece of code in R. And at the end of this session, you can download this code and run it on your system as well. So what I will do is I will go into this lesson 6. Okay, this is the link. And we'll just go through this lesson with a little exercise at the end. Okay, so please let me know once you're on this link. We're going to take just a few minutes. It's not going to be a long time. All right, perfect. So here we have a different problem. There, we had multiple samples. And the question was, how do these groups of samples compare to each other? In this case, we will just take one sample. So we don't have anything to compare to. We just want to understand what's in it. And to do that, we will use a different pipeline. So this is a pipeline that is going to do something very different. Here we will take full 16S ribosomal RNA gene that is sequenced. We're going to align it to a reference database of all 16S ribosomal RNA genes. We're going to count how many do we have of what kind, and we're going to annotate them. Okay, and so here, the basic table that we're going to be dealing with looks like this. You have individual elements, genomic elements, for example, 16S ribosomal RNA, and you can see how the kingdom, phyla, etc., is organized according to the different taxonomic classification with how many of each kind do I have. And then you will also be able to annotate, right? So separate them and see how many do you have of different types of, for example, phyla, right? So if we just focus on phyla, we can see how many of each kind do we have. Now, they will come up many times, right? Because multiple organisms fall under different phyla. So we need to combine them together. So after we combine them together, we can calculate percentages. What's the percentage of a particular type from the total that we have? And obviously we can visualize such data, right? We can prepare some kind of a visual representation, and that is sometimes more informative. We can do a pie chart, or we can do a bar plot. So essentially we can see what is happening with our data. And so this lesson is to be able to do these simple steps in R. If you already know R, you know that for many different applications in R, there's typically a library that already contains many of the functions that you would like to use. So you don't actually have to reinvent the wheel. Some of these packages are very specific, like phyloseq. They just produce a set number of plots that are useful for a particular application, like, for example, phylogenetic analysis for phyloseq. But some of them are more general libraries, and those libraries include ways to visualize data and create different plots, or maybe calculate different statistics for different types of things that you can find. So here we'll do something very simple. We'll learn how to load our data, prepare a pie chart, and then we'll make a bar plot, because bar plot could actually be more easy to interpret visually than a pie chart. So the objective would be just to understand how many of each kind do I have in a particular single sample without having to compare to anything. So here we have some code, and let's just go through this code really quickly. We load a library. This library is to load an Excel file. Now, sometimes you will have CSV files, TXT files, Excel files. So you need to be able to load those data and load them into your environment, into your script. Here, we'll do it right here on our server, but you can also do it in R. So then we want to sort the data. And sorting the data is useful because instead of looking at images that look like this, we can kind of organize them to all look like this, from highest to lowest. And once we do that, we then create percentages, make a pie chart, and also make a bar plot at the end. So let's see how difficult is it for us to perform all of these steps, including adding some color here as well. Okay, so here you have a console, which is where you run the code. And in this console, you already have all of the data and all of the script loaded in. In fact, if you want to use this data set on your computer, you might want to download it right here. So just right click on this link and download linked file. If you want to see what happens when you run this code should run in exactly the same way on your computer as it does here. The only thing that you need to remember is that we're using a library. Yes, you can run it on RStudio. But in RStudio, you will need to have, the only library that we're using is this additional library, Excel, to read Excel files. Okay, so let's try to run it. And you can see that, first of all, we have a success, right? We correctly have the answer. And right here, we have the results. So this is a pie chart that we created, and this is a bar plot that we created. And this bar plot is already annotated. Now, let's try how to change just one little thing so that we know that this is actual code that runs and not just an automatic button. So one of the things that you can see here is that when we create a pie chart or a bar plot, we actually give them a title. Main is a title. And here we also make different dimensions for our plots. So we're going to do two things. We're going to increase the dimensions because this is kind of small. And we're actually going to change the title. Okay. So here we're going to change this to pie chart. And here we're going to add bar plot. And you can try to do this in RStudio as well. Okay, so here you can see that we also add the label to our x-axis. And so when we look at the x-axis right here in the bar plot, You can see bacteria type. Okay, but actually the type is by phylum, right? So here we want to add phylum. Okay, now once we do this, oh yeah, we also wanted to increase the size. So let's do it like this. And let's run it again. Okay, so here, pie chart, right? We changed that. And here, we also changed phylum and bar plot right here. And I noticed, by the way, that I capitalized the C so we can make it smaller. So this is an actual piece of code. If I want to take it to my computer, what do I need to do? Copy, right here is the copy code button. Open RStudio. If anyone doesn't have RStudio, we have a little tutorial on how to install RStudio. And then you can run the exact same code. Obviously, you would need to set up your environment to have the library and to have the file. Okay, so here's our RStudio. Let's make it bigger. Create a new script. Paste it here. Okay, and here, right, we have this library. I already have it installed. Let's run it. Okay, object labels is not found. So right here we have, so I think, what is happening? Let's run it like this. Okay. Like this. And run it like this. All right, and so there we have it. It also works in RStudio. Okay, so to summarize, because we're already going over time, let's wrap this up and summarize what we have done today. The objective of this program is to provide us with methods of analysis that we know how to perform and we know how to interpret. Today, we ran a demo pipeline called the data-to pipeline, and we saw this data-to pipeline in terms of the inputs and the outputs, kind of understanding the logic of analysis. Now, there are many other types of analysis that we can do, and we looked at this annotation of 16S ribosomal RNA. We have the kind2 pipeline as well, and we have this data-to pipeline. So we can look at those different examples and how they approach the same problem with different other methods. We also looked at this simple example of visualization. How can we visualize the data that we have from sequencing? And we saw that we have this abundance of proteobacteria in this particular sample. Now, what do you do moving forward? So I recommend several different publications that you can go through to kind of get started moving further into this field of metagenomics. There's a good review of the Human Microbiome Project, which contains a lot of 16S ribosomal RNA data used in a variety of studies. You will also find here a paper, I'll share the links in just a moment, bioinformatics for microbiome research, concepts, strategies, and advances, and also explore the actual human microbiome project as well. So this is a collection of data that has 16S as well as metagenomics as well. And you can see different types of collections within this project. So let me get you these links real quick. Okay, so here are those links. Okay, now moving forward inside the actual tutorials, you will find these different additional resources. An overview of sequencing, methods of analysis, different project examples, including some exercises that you can do. And then hopefully after you complete that kind of training, you will be able to apply this to a data set of your own. So we do have a whole project where you can go through several lessons on how to develop an independent research project, including where to find data, how to plan for your analysis, and if you need help, how to submit it to some of our mentors to give you advice and direction on how to develop an independent research project with omics data and bioinformatics.